Hello everyone and welcome to Pocket Size Bio with Dr. Casco. In today's video we're going to continue Chapter 9, Cellular Respiration and Fermentation. We will start from section 9.4, which is during oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthesis. So let's get to it. Let me just move the notes, zoom into the notes a little bit. All right. Okay, continuing on from the previous sections, we talked about glycolysis, we talked about the Krebs cycle and how the Krebs cycle uses the process where you essentially harness all the electrons from glucose and you reduce NAD plus to NADH. We also produce a little bit of ATP from glycolysis and the Krebs cycle. You're producing also FADH₂ and so forth. Ultimately, to shuttle electrons to the electron transport chain, as we're going to see in this particular section. Okay, so looking at the notes here, so far we have 4 ATPs being produced, right, during respiration, okay, from substrate-level phosphorylation, okay, substrate-level phosphorylation. Now, NADH, again, as I just mentioned, NADH and FADH₂, they will account for the most energy extracted from glucose. Okay, we haven't seen the massive ATP production yet, but here's where we're going to see it. Okay, so the NADHs are what link glycolysis and Krebs cycle to the oxidative phosphorylation process. So we're going to see this process involves the inner mitochondrial membrane, okay, as it has the proteins embedded on its on that inner membrane, okay, for ATP synthesis. So, and by the way, I'm going to be using the word ET or the abbreviations ETC, right? Electron transport chain, ETC. You're going to see that throughout my notes, but just, keep in mind that I'm referring to the electron transport chain. Okay, and the electron transport chain is embedded within the cristae, which is the inner mitochondrial membrane. Okay, something to also note as we look deeper into that structure, right, of the mitochondria. Of course, the inner membrane, as we've seen from the tour of the cell chapter, there's a lot of surface area, right? There's a lot of folds occurring within the inner membrane, and to increase surface area, so you can have more complexes of the proteins embedded within the cristae. So you can be producing more ATP in a given period. So you have many, many complex copies, if I can say. All right. So this ETC is composed of, proteins, or the majority of it is proteins that are in complex, right, with other proteins, and you have this whole system, as you can see here in the figure. And these complexes and these proteins, they're aggregated together to create these complexes, and there's 4 major complexes, as you see here. So here's a protein complex 1, complex 2, complex 3, and so forth. And they all work together with other molecules or other compounds and chemical groups that are referred to as prosthetic groups. And these prosthetic groups is essentially cofactors or coenzymes that are working alongside with these complexes. Okay, and ultimately what happens here is that electrons coming in from NADH, they're going to drop electrons into this complex, and you're going to have a cascade of electrons passing from one complex to another, to another, to another, to another, and to another. Ultimately, where the last acceptor of these electrons is oxygen. Okay, so oxygen will accept these electrons, use hydrogen plus ions that are in solution to create water. Okay, each chain component is reduced by its uphill neighbor. We're going to see this in a little bit, and oxidized by its downhill neighbor also. Ultimately, what you need to understand is that electrons are being moved, channeled from one carrier to another, to another, to another. So they're always in this play between a reduced state and an oxidized state, reduced state and an oxidized state. So in this, in the beginning here, you have NADH. It's in a reduced state, right? And that NADH will transfer its electrons to the first molecule of the cascade, or the first within complex number 1, what is called a flavoprotein. And you can see here we begin this kind of cascade. NADH will come, it will provide the electrons, it will become oxidized, NAD. This will go back to the Krebs cycle to be reduced again and then come back. Okay, so here it provides the electrons to the flavoprotein, which you see FMN here, and this just means that it contains this chemical group, flavomononucleotide FMN. Okay, this is a molecule you should remember just because it's the beginning of the cascade, FMN here. And then this one will then get oxidized, reducing your next molecule, which is an iron, a protein containing iron and sulfur. Okay, then this one will then further get oxidized, reducing your next compound. And I'm just going to highlight this for emphasis. Ubiquinone. Okay, this is a very famous protein here. It's designated by Q. Okay, it's also known as coenzyme Q. Now, this is a hydrophobic molecule and is a non-protein. Sorry, so the only non-protein member. So it's a hydrophobic molecule, and it's actually it moves around. It kind of shuttles from one complex to another, as you're going to see in the later pictures. It has some flexibility in movement here. Ultimately, this further, goes into the cascade. Ubiquinone will pass electrons into another complex, okay, complex 3. So it can receive electrons from complex 1 and complex 2, right? Ultimately, it will shift and move electrons into complex 3, okay? Again, the idea of that it can shuttle and move around is mobile within the membrane. We're going to see that in a little bit. Okay, and then the remaining complexes, the majority of the proteins are known as cytochromes. Okay, cytochromes meaning that they're proteins that have heme groups, okay, within their structure. And heme groups, you have these chemical groups that contain iron. Okay, similar to the heme groups in hemoglobin. Okay, so they contain iron. So you have this cytochrome B. Again, passes the electrons to another molecule, another complex and protein, flavoprotein group. Again, having iron and sulfur. Not flavoprotein, sorry, an iron sulfur protein. Okay, and then you go to more and more cytochrome proteins there. Ultimately, and this is probably one that you need to remember. If you're an AP, you don't need to memorize every single detail here. Maybe just the beginning, ubiquinone and cytochrome A3, which is the last carrier before passing it down to oxygen. Okay? So cytochrome A3. Each of them have their distinguishing characteristics that maybe goes a little bit beyond the scope of what you need to know, even for undergraduate biology. Okay, so ultimately, this cytochrome A3 will pass electrons to that oxygen. Since oxygen has the highest affinity of electrons, it will accept them with hydrogen plus ions. You have these two protons to produce water. Okay. Electrons coming in from FADH₂, which, remember, it's also another kind of shuttle molecule, or an electron carrier. It also provides electrons, but at a lower energy level. Okay, has a lower free energy and starts at a lower level. So this one comes in from complex number 2. So these two come in. They ultimately come in from complex 1 or 2, and they provide electrons. These electrons get shuttled down from one protein to another, to ubiquinone. They further keep going down the cascade, ultimately going to oxygen. Okay. Remember, in the electron transport chain, FADH₂ comes in from complex number 2. Now, if you notice, there's no ATP synthesis yet. Okay, they're just providing a way for electrons to lower their kind of energy, the free energy that they have. They're liberating that energy one little by little in manageable steps. We're going to see now how and in what way ATP is being produced. So let me just move this, adjust the zoom here. Okay, now we go into chemiosmosis. Chemiosmosis couples electron transport and energy release to ATP synthesis. Okay, very, very important protein complex that you need to know is ATP synthase. Okay, ATP synthase is found embedded within the cristae, within the inner membrane, and this is the actual complex that will create the ATP. Okay, this is the actual one that will drive the reaction of ADP plus inorganic phosphate to produce ATP. Okay, and it's quite incredible how it looks like. It's a rotor, a motor, that is pumping, hydrogen plus ions to drive ATP production. Okay, so it does work like an ion pump running in reverse. Okay, it's not consuming ATP and moving substances from a low to high concentration. It is producing ATP from a concentration of substances going from a high to low concentration, in this case, hydrogen plus ions. Okay, so we know enzymes can catalyze a reaction in either direction, depending on the delta G, where you have that free energy. But in this particular case, under aerobic respiration, because of the electron transport chain, you are getting also hydrogen plus ions being pumped. Okay, I'm jumping a little bit ahead of myself, into the intermembrane space. Okay, between the inner and outer membrane, you're getting hydrogen plus ions being pumped out into this intermembrane space. So what's happening is that you're creating a concentration gradient. Okay, you're creating high concentration of hydrogen plus ions. Okay, as you see here. I'm going to go back a little bit just to not go ahead of myself. Okay, but you're going to understand now what I mean by these ion gradients. So under respiration condition, ATP synthase uses this ion gradient to power ATP synthase. Okay, and more specifically, this hydrogen plus ion gradient. Okay, so the power source is a difference in hydrogen plus ion concentration between these two spaces here. So you're having good potential energy in this particular area. Okay, and when you have enough potential energy and you have this concentration gradient, you have enough energy to drive cellular work, in this case producing ATP. Okay, so this process where you're having a gradient to drive cellular work, that is called chemiosmosis. Okay, osmosis in the sense that it's referring to a flow of hydrogen plus ions across a membrane. And that flow of these ions is what drives the ATP generation. Okay, so this is how ATP synthase looks like. It has several parts. You have something called a stator. You have a rotor in this particular area here. Let me switch here. So you have your little stator is a stationary unit of this protein complex. You have this rotor area here. And this, just as the name says, will change shape and spin about in a central axis. Okay, this axis is called the internal rod. And then you have an area called a catalytic knob where the actual ATP synthesis is occurring. Okay, so there's four main parts there. All right, four main parts. You have your stator, rotor, internal rod, and the catalytic knob. Okay, and so the protons that you see here, the hydrogen plus ions, these ions, they move ultimately into binding sites on the rotor. And when they bind to this rotor, they force it to spin about in its axis. And that spin, that mechanical movement, will help catalyze this ATP production that you see here. Okay? ATP synthase is the smallest molecular rotary motor known in nature. That's quite fascinating that this is happening at such, a molecular scale. We can see this type of mechanics occurring. Okay, now AP students probably don't need to know this level of detail. Undergraduates probably do. So I'm just going to go over this and read this through for you guys so we can see it together. So how this actually works, you have this gradient. Hydrogen plus ions flow through, or they enter through down their gradient through a little channel in the stator that you can see here. And this is anchored directly to a membrane. Just like mechanical things that we use of a motor, you have this stator here, which is a stationary component of your motor. Okay, so these hydrogen plus ions will come in, pass through these channels, and bind to particular areas within the rotor. Okay, and they cause a shift, right? A change in shape in the rotor that would enable the rotor to spin within the membrane. So all of this is embedded within the membrane. Okay, so these ions will, as they bind, they'll be moving and shifting and moving and moving and moving and moving until they make a whole complete turn. When they reach back to their original place, they will exit through another little channel here and be released into the mitochondrial matrix side of the mitochondria. Okay, and the matrix side. As they pass down, they enable this internal rod, okay, to also spin about. Okay, and this spin will enable will catalyze the reaction between ADP and inorganic phosphate to produce ATP. Okay, so turning of the rod activates catalytic sites in the knob that will produce ATP. Okay, all of this is occurring in the matrix side of the mitochondria. Okay, not too difficult there, but just to give you a sense of how it works. Okay, so let me just zoom in here. All right, so the electron transport chain, the ETC, generates and maintains the hydrogen plus ion gradient. So this is what you really need to know. Okay, so as we're getting electron flow, exergonic electron flow from NADH into this complex, right? Hydrogen plus ions will be pumped into the intermembrane space. Okay. Hydrogen ions, they have the tendency to diffuse naturally down, just natural diffusion from its gradient. Okay, so ATP synthase molecules are the only place, and this is quite interesting, this is important, it's the only place where hydrogen plus ions can diffuse back to the matrix. So they have to pass through this motor, right? This will improve efficiency in creating ATP. Okay, the exergonic flow of hydrogen plus ions is then used by enzymes to generate ATP. So this coupling, this use of hydrogen plus ions to change binding sites, to activate binding sites in these in this catalytic knob area, is chemiosmosis. Okay. So looking at the complex in itself of proteins, certain electron carriers, they can accept and release hydrogen plus ions as you're having this cascade occurring. Okay, so you're forcing hydrogen plus ions to go from one area to another. Electron transfers at certain steps. Electron transfers at certain steps can cause hydrogen plus ions to be taken up and released on the other side. Okay, this highlighted area is probably the most important that you need to know, right? Electron carriers are arranged so that protons could be accepted from the matrix and deposited into the intermembrane space, and that resulting gradient that you're creating, that is being created, is the proton motive force. Okay, having good potential energy, having the capacity to do work. Okay, they refer to it as the proton motive force. Okay, so it's this force that drives hydrogen ions back through the ATP synthase channels that can enable ATP to be produced. Okay, proton motive force. All right, let's continue. This is the same image that I have placed in the previous page, just for you guys to see it in a little bit more detail there. Okay, chemiosmosis is an energy coupling mechanism using a hydrogen plus gradient to drive work. All right, so this whole idea of of creating this gradient of hydrogen plus ions ultimately comes from the exergonic redox reactions, right? The reduction oxidation reactions that we've seen before in the Krebs cycle. Ultimately, to have the capacity to harness that energy and produce that proton motive force, it drives ATP synthase. Okay, not only the mitochondria does that, the photosynthesis and chloroplasts also do that too. Chemiosmosis in chloroplasts also generates ATP in the light reactions that we're going to see in chapter 10. In this case, light drives electron flow and gradient formation, okay, in the thylakoids. We're going to take a look at that later, so don't worry about that for now. Okay, and not just chloroplasts, prokaryotes. Remember, they can also produce ATP in a very similar way. They generate hydrogen gradients across their plasma membrane, and they have this they produce this proton motive force to generate ATP, to pump nutrients, to use, for any active transport or just activity that you need, rotate flagella to move about, and so forth. Okay. All right, let's continue. Accounting of ATP production. So, in a nutshell, we're almost done with this whole process of cell respiration. Okay. Let me zoom out. Okay, energy flow. Now we've seen the entire process in itself. We know that glucose, we come in, we obtain glucose after eating, we process that food, break it down, we have our molecule of glucose. The glucose will slowly be broken down, oxidized, stripped from electrons. Ultimately, these electrons are passed down to energy electron carriers, NADH primarily. These NADHs will transfer electrons to the electron transport chain, producing a proton motive force, basically a hydrogen plus ion concentration gradient, which will then be used to produce ATP. Okay, so this you need to be able to explain by yourself. Okay, make sure you understand enough that you can explain the process and the importance of each process, right? And ultimately to produce ATP. Okay, we know 4 ATPs are being produced by substrate-level phosphorylation. Ensure that you know that idea, where and how is ATP being produced, substrate-level phosphorylation. Each NAD contributes enough energy to produce, to generate a maximum of 3 ATPs. Okay. Now, the exact number of ATP production, it's a little bit difficult to calculate, and there's several reasons for that. The ratio in which you have NADHs carrying these electrons to produce ATP is not really a whole number, right? One ADH technically can pump out 10 hydrogen plus ions. But studies have shown that you need 4 hydrogen plus ions to generate 1 ATP. So 4 hydrogens, that's 1 ATP, another 4, that's 2 ATPs, and then you have 2 hydrogens left, you're gonna have 2.5 ATPs. Okay, so it isn't a fixed number. You have some variation there. Number 2, ATP yield can vary, okay, by the shuttle used from electrons that are coming in from the cytosol. What do I mean by this? FAD being the electron carrier, or NAD plus being the electron carrier, can give different things, different amounts. And the cells can also, they use different shuttles, can yield different numbers, right? So in brain cells that primarily uses FAD as its shuttle, yields 2 ATP, okay? In liver and heart cells, you have NAD shuttles that can yield 3 ATP, okay? As they're using that more than FADH. So you can have variation. That's what I'm kind of trying to state here, okay? So this is another factor that can yield the net difference there. And then the proton motive force, okay, that hydrogen gradient, can be used to drive other things, not necessarily the ATP production. It could be used to reabsorb or reuptake pyruvate, so you can use it for something else maybe. Okay, so that will lower the possible yield that you could have of ATP production. Okay, so these are reasons why you can have fluctuations between per glucose molecule and so forth. But in itself, if everything goes well, there's no external things that are changing the output of glucose, okay, you will get 28 ATP from oxidative phosphorylation, basically the ETC and ATP synthase, and then you'll get 4 ATP from substrate-level phosphorylation. So ultimately, you'll get around 32 ATP in total, okay? Depending if you're using different things, you can get 30. So in your textbook and in different books, you might see a range between 30 or 32 ATP, okay, per glucose molecule. A complete oxidation of glucose would release 686 kilocalorie per mole, okay? Phosphorylation of ADP requires 7.3 kilocal per mole. So you can actually calculate the efficiency in which you are, using the oxidation of glucose to produce ATP. In this case, it's called the respiration efficiency. It's about 34% that you can extract energy from a glucose molecule, okay? 34%, which is pretty good in terms of harnessing energy. Actual percentage is probably higher under cellular conditions, but we tend to go for this number, okay? The rest of that energy, harnessed from glucose is ultimately, the process through glucose is lost as heat. Okay, cell respiration in itself is pretty efficient. Interestingly, a car doing combustion is only 25% efficient. So that's quite interesting there. Under certain conditions, reducing the efficiency could be beneficial, and you'll see this more, in animal behavior. Hibernation, right? We know this is a time where organisms, they get into a very low activity, or enter a low activity state. A low metabolic activity is occurring. Mammals have brown fat that is packed with mitochondria, and even in this particular condition, you're going to lower the amount of ATP being produced, just because you want to optimize the minimal as possible, right? Okay, so the inner mitochondrial membrane has an uncoupling protein that basically allows protons to flow back without generating ATP. Okay, so hydrogen plus ions will go naturally back down without necessarily generating ATP there. Okay, you don't need to generate a surplus of ATP if it's not necessary. Okay, so activation of these proteins results in oxidation of fats. Okay, generating heat without ATP production. And of course, remember, you want to have certain gradients. If you don't have this and you're not using ATP, you're going to have a surplus of ATP buildup, and it's going to shut down the whole process of respiration. And you also don't want that to happen. Okay, so this is a way to maintain an organism in a hibernating mode without deterring the metabolic processes greatly. Okay. All right. So now we're going to get into, kind of finished aerobic respiration. We're going to go into fermentation and anaerobic respiration. And this enables cells to produce ATP without the use of oxygen. All right. So without oxygen, oxidative phosphorylation will cease. You don't have oxygen, you don't have that last acceptor for electrons, everything will, in this system will stop. So what happens? There's two mechanisms that can oxidize fuel and generate ATP without oxygen. It's fermentation and anaerobic respiration. Okay, there are two different things. So an ETC is present in anaerobic respiration. So you still have the electrons transport chain being used, but in fermentation, in that fermentation process, it is completely absent. Okay? So let's talk about anaerobic respiration. Anaerobic respiration, again, uses the ETC, but you don't necessarily need oxygen to be the final electron acceptor. Certain bacteria can use sulfates as the last electron acceptor, ultimately producing hydrogen sulfide. Okay, so, and you can see these, you can smell these, let's say on swamps or where you have that sulfur smell. You have these particular bacteria that are using sulfate as the last acceptor here. Okay, and anaerobic respiration. Still, the electron transport chain in the cascade is working. ATP is being produced, but you don't need oxygen. So remember that. Fermentation. Fermentation oxidizes organic fuel and generates ATP without oxygen or the electron transport chain. So you don't need anything of that. You don't need oxygen. You don't need the complexes of proteins on the membrane. Nothing. Okay, what happens here? Glycolysis. So you're focusing primarily on glycolysis. Glycolysis oxidizes glucose to pyruvate. That's the same, with NAD as the oxidizing agent. Okay. Glycolysis, we know is exergonic. It produces 2 ATPs, okay, by substrate-level phosphorylation, and you don't need oxygen for it. Okay, there's two types of fermentation, depending on the organism, depending on what pathway you get into. Okay, and fermentation, I'll go a little bit higher. I'll come back to this little diagram here. Fermentation allows ATP to be generated from glucose, again, by substrate-level phosphorylation. And moreover, fermentation enables the recycling of NAD. Okay, this is probably what's more important. NAD by transferring electrons from NADH to pyruvate or derivatives of pyruvate. I'm going to explain what that is. Okay. Aerobic conditions, remember that generally NADH will go to the ETC to give the electrons, and then it'll turn back into NAD+. Then it'll go to the Krebs cycle again to be, regenerated and regenerated. But since we don't have oxygen for that to occur, okay, they get regenerated by these two processes. So there's two types of fermentations. There's several types of fermentation, but the most common in textbooks is alcohol fermentation and lactic acid fermentation. Here, essentially, you get pyruvate. We know pyruvate now. This is a process that will convert pyruvate to acetaldehyde, which will remove a carbon dioxide. So you're going, and I'm going to just maybe zoom out a little bit, okay, where you have a 3-carbon compound, okay? So you have a glucose undergoing glycolysis. Remember, we produce the pyruvate here, a 3-carbon compound, okay? ATP is being produced in glycolysis, and then this pyruvate will undergo will be converted into 2-acetaldehyde. Remember, you have 2 molecules because you're working twice. One molecule of carbon dioxide will be created, so you're losing a carbon per molecule of pyruvate. So here's 2 and 2. So you have a 2-carbon compound. Okay, and then this acetaldehyde will then be converted into ethanol via the acceptance of electrons and hydrogen plus ions. Remember, you produce NADH in glycolysis, but since you can't use NADH for the ETC, for the electron transport chain, it has to get oxidized somehow so you can continue producing ATP. Okay? This is called NAD plus regeneration. So acetaldehyde will be reduced to ethanol. Okay? Alcohol fermentation reduces acetaldehyde to ethanol, ultimately regenerating NAD+. Okay? So you have certain organisms doing this. Yeast is of particular importance. We all know we use yeast for commercial use, for in industry, ultimately for brewing, creating alcoholic beverages, baking, winemaking, etc. Okay? Alcohol fermentation. No need for oxygen. Okay? Just glycolysis. Another form is lactic acid fermentation, in which pyruvate is reduced directly by NADH to form lactate. Okay? So here you have this particular situation, lactic acid fermentation by some fungi, by bacteria, and we use these. There's, industrial applications for this to make cheese and yogurt and whatnot, right? Humans also can undergo this particular process. Human muscle cells can switch to lactic acid fermentation when oxygen levels are scarce and there is not enough oxygen to reach all the cells in the body, particularly in muscles, when you're doing strenuous, very intense exercise. Okay. Lactic acid was previously thought to cause muscle fatigue. Okay. But potassium plus ions may be to blame, right? Lactate may enhance that performance of muscle and ultimately cause that fatigue, right? We're going to see this in a little bit later. Excess lactate, when you're producing this lactate, can be carried in the blood and processed in the liver to be converted back into pyruvate. So you can, even cycle this back to pyruvate. Okay. And then which can ultimately enter, be processed in the mitochondria, and then you can just do normal cellular respiration. Okay. What you see here, again, the visual for lactic acid fermentation, same idea. ATP is being produced in glycolysis. NADH is being reduced, NAD plus is reduced to NADH. Pyruvate then gets reduced into lactate. So you can, again, regenerate NAD plus. Let's continue here. Okay. Almost reaching the end here, comparing fermentation and cellular respiration, right? This is a good place to put a little star or to note down to get the core ideas on differences between fermentation and cell respiration. This is a very common question in AP or an undergraduate level, critical thinking, compare and contrast fermentation and cell respiration, right? So one, fermentation, anaerobic respiration, aerobic respiration. There are three pathways in which you can produce ATP from sugar. So in that sense, they all do something similar. They produce ATP. They are able to reduce NADH. Okay? They all include substrate-level phosphorylation. You produce 2 ATP. You produce pyruvate, okay? And NAD plus is the main oxidizing agent, okay, that accepts electrons from food. Now, in that critical difference here is the mechanism for oxidizing NADH to NAD plus. So again, regenerating NAD plus. For fermentation, the final electron acceptor, okay, is an organic molecule. Now we know it could be acetylaldehyde or the lactic acid, the lactate. Okay. For cellular respiration, you have NADH electrons that will be transferred to the ETC, where the final electron acceptor is oxygen to regenerate NADH plus. And I say oxygen, we know that it's FMN, it's the first, that flavoprotein is the first one, but ultimately, if you don't have oxygen to accept the last electron, you're going to stop the cascade of flow of electrons. So it's, you're gonna stop that relay of electron flow. So for electron respiration, for aerobic respiration, you have oxygen. For anaerobic respiration, you have sulfate. Okay, so make sure you understand that. What are key differences? And the point is regenerating NAD plus. Okay, regenerating NAD plus. So, and some of these final notes here, more ATP is produced by the oxidation of pyruvate in the mitochondria that is unique to respiration, aerobic respiration. So I've mentioned this before, but just to kind of give you that idea, right? Without the ETC, the energy in pyruvate ultimately is unavailable. Cell respiration will give much, much more energy per sugar molecule compared to fermentation. Aerobic respiration will yield 32 ATP per glucose. Fermentation will yield 2 ATP. Okay? Very, very important. Compare and contrast, okay? So by the nature of these different processes, anaerobic respiration, fermentation, aerobic respiration, different organisms, they vary in the pathways that they use and to break down sugars, and they have different terminology depending on what pathway they use. Ultimately, if they use alcohol, if they use oxygen or not. You have organisms that are referred to as obligate anaerobes that carry out only fermentation or anaerobic respiration. Okay? So there's no use of oxygen, and actually they cannot survive in an oxygen medium. Okay, they will die. Some cells, like vertebrate brain cells, can carry out only aerobic respiration. So they're strictly aerobic. Okay? You have to have a continuous supply of oxygen in these tissues, brain tissues. Okay? Yeast and many bacteria, they could be facultative, anaerobes. So they can survive using either fermentation or respiration. They could use oxygen. Okay? Human muscle cells can behave as facultative anaerobes. They can use oxygen when it's available. They can use fermentation when it's available, when it's necessary. Okay? And for facultative anaerobes, pyruvate is the key point in which it is decided either you'll go into aerobic respiration or any type of anaerobic process. Okay? Pyruvate, we know, is converted to acetyl-CoA if oxygen is present. If not present, you can have ethanol, lactate, or other forms of respiration, depending on the organism. Okay? Ultimately, what's important is that you recycle that NAD plus. Okay? To make the same ATP, this is a good thing to point out, a facultative anaerobe must consume sugar much faster when fermenting. Of course, you're producing so little, so in order to kind of get that same yield, you're going to have to be breaking down sugar at a higher rate. Okay? Talking a little bit about evolution and glycolysis. This is very, very interesting. Ancient prokaryotes, it is thought, most likely used glycolysis to make ATP before oxygen was even present in the atmosphere. Oldest bacterial fossils are over 3.5 billion years old, before oxygen even accumulated enough in the atmosphere. So they had to do an anaerobic process. Cyanobacteria produced oxygen as a byproduct 2.7 billion years ago. So these organisms, these first ancient organisms, must have generated ATP exclusively from glycolysis. So glycolysis must have been that first metabolic pathway to produce ATP. The reason why we say evolutionary basis is because glycolysis is ubiquitous, right? It occurs in the cytosol. It occurs in almost all organisms. So it is thought that it evolved very early, and it's still found today, right? It does not require organelles, enclosed organelles. So that's incredibly interesting. And organelles evolved 1 billion years after the first prokaryotic cell. So this process has been around, and it's so effective that it's still around today. So this gives a sense, one, it provides evidence of consistency within metabolic processes, and also how we're related at a biochemical level. So that's quite interesting, right? Very, very interesting, and also a kind of a piece of evidence for evolution. Okay. So last thing we're going to talk about, we're in section 9.6. Glycolysis and the citric acid cycle connect to many other metabolic pathways. Just zoom in here. Yes, so glycolysis and the citric acid cycle, if you remember, if you've seen my previous videos, I showed an intersection diagram of many pathways interconnecting together like a train system, right? And that's what we see here. Okay, it could serve glycolysis and the citric acid cycle, the Krebs cycle, can serve as intersections for catabolic and anabolic pathways. You usually don't find free glucose readily available for humans in this single form, just a glucose molecule floating around. And we generally get most calories from fats and proteins and carbs in the form of sucrose. Okay, and you can use these different molecules to produce ATP, right? Not strictly glucose only, right? And ultimately, you'll break glucose into that pyruvate and into to produce ATP. So glycolysis in itself can accept a wide range of carbohydrates, okay? Where you can initially start with polysaccharides, starch, glycogen, and so forth, which can ultimately be hydrolyzed into glucose monomers. If you remember from chapter 5, how we saw this amylopectin and amylose and starch and glycogen, they can break off glucose monomers from the tips of these branches, right? And digestion of disaccharides, sucrose, can provide glucose and other monosaccharides, fructose. Okay, so you can get the glucose from different sources, right? Proteins and fats can also enter respiratory pathways. Proteins, they could be broken down into their amino acids, and they can be converted into pyruvate. They do need to be processed. You have amino acids and you have a lot of nitrogen groups. Of course, in the amino group, you remove these amino groups in a process called deamination. Okay, and this deamination will remove these amino groups, which will then be converted into nitrogenous waste to be excreted out of the body, like ammonia and urea. Okay, as you see here. All right, so again, fats can be broken down. Fatty acids can turn into acetyl-CoA. Glycerol can be used to turn into glyceraldehyde 3P, G3P. And amino acids, as I mentioned before, can be also used as a pyruvate, acetyl-CoA, or directly into the citric acid cycle. Okay, in terms of carbon skeletons, so not just to be used, for ATP production, but also as a molecule in the process, right? So catabolism the carbon skeletons are modified to intermediates of glycolysis and the citric acid cycle. Okay, so catabolism can harvest energy from fats. Fats can be digested, as I kind of showed in the diagram before, broken down to glycerol and fatty acids. And then the glycerol can be converted into G3P, okay, that first initial 3-carbon molecule, okay, in glycolysis. Fatty acids can be turned into 2-carbon fragments, via beta oxidation. And these fragments ultimately can turn into acetyl-CoA, enter the Krebs cycle. Okay, and then you have ultimately, the regeneration or the production of NADH and FADH₂ to enter the electron transport chain. Interesting, a gram of fat oxidized by respiration can generate twice as much ATP as a gram of carbohydrates. So they're more energy-rich molecules. You can yield more ATP, okay, compared to carbs. Okay. In terms of anabolic pathways, remember, so we talked about catabolic pathways. Complex molecules are broken down to produce some intermediate within the whole cellular respiration sequence. And then you have biosynthesis, anabolic pathways when you want to create things. So metabolic pathways can help produce more complex molecules. And in this case, food generally provides carbon skeletons for the cells to make these molecules. And some organic molecules are used directly from digestion, but you can get intermediaries in glycolysis, and the citric acid cycle can be diverted. I'm going to highlight this, where these intermediate molecules, right, can be used to produce more complex molecules. Okay. A human cell can synthesize half of the amino acids, in the case for polypeptides, half of the amino acids just by altering citric acid cycle compounds. So you can extract intermediates from the Krebs cycle to be used to create other things like amino acids. Okay. Other amino acids definitely we need to be getting from diet. So we cannot produce all of them. A good portion though. Okay. Glucose can be synthesized from pyruvate. Fatty acids can be synthesized from acetyl-CoA, right? So just as we're using these molecules to produce acetyl-CoA, we can reverse the process. Pyruvate can go back to produce glucose. Acetyl-CoA, a very small molecule, can be used to produce fatty acids. Okay. Anabolic pathways do not generate ATP. So that's, of course, you're creating more complex and more complex molecules. You're going to have to use ATP, right? So they consume ATP. Okay. So glycolysis and the citric acid cycle, Krebs cycle, can enable cells to bounce around depending on the needs of the cell. Okay. Excess carbohydrates and proteins can be converted into fats. So this is quite interesting, because if we eat more food than we need, we will store it as fat, even if your diet is completely fat-free. Because of this interplay between different molecules, you can produce ultimately fat. Okay. So that's quite interesting there. And the last thing we're going to see for this chapter is the regulation of cell respiration via feedback mechanisms. We've talked about feedback mechanisms, feedback inhibition during enzymes, okay, in the chapter on enzymes, chapter 8. We're going to talk a little bit about it here. So, supply and demand regulate metabolic economy. And the cell is very, very good at regulating its concentrations of certain molecules in general. Excess of an amino acid can trigger feedback inhibition, okay, to prevent overproduction, a surplus, okay? And we know this as negative feedback mechanisms. So what happens here, and I might just change this, give me a second here. Okay. The end product that is being produced in this whole process will inhibit an early enzyme in its particular pathway, preventing diversion of intermediates. Okay. The rate of catabolism is regulated. Low levels of ATP will speed up the process. If you have an abundance of ATP, then you're going to slow down the whole cellular respiration process, because if you have an abundance, why are you going to produce more and more and more? You're just going to waste energy and materials. So our cells have this feedback inhibition mechanism. Okay. Control is based on regulating enzyme activity at very strategic points. One strategic point is catalyzed by phosphofructokinase. Make sure you remember this enzyme. Okay, phosphofructokinase, it catalyzes the earliest step that irreversibly commits substrate to glycolysis. So this particular enzyme functions as a pacemaker for respiration. Okay, and the way that it works, and I'm going to show you the diagram really quick, is that it is an allosteric enzyme. And these allosteric enzymes, if you remember, they have receptor sites and they can bind to several complexes and several substrates. It's inhibited by ATP. Inhibited by ATP. Okay. It is stimulated by AMP. Okay. AMP is adenosine monophosphate, so it's only one phosphate. Okay. And how is this AMP and the stimulation occurring? High ATP levels will slow glycolysis down. Okay? If you have low ATP levels, that means you're consuming ATP and you're producing ADP. And then further down, the body will produce AMP. So these concentration differences will stimulate phosphofructokinase. Okay? Citrate, the first product of the citric acid cycle, can also act as an inhibitor alongside with this phosphofructokinase. Okay, so this synchrony between the levels of ATP and the cellular pathway, respiration pathway, are regulated by these structures, these molecules, these enzymes, right? Citrate and this kinase here. Let's take a look at this diagram. Okay. So in glycolysis, you have glucose coming in, it will produce fructose 6-phosphate. And then this enzyme, if you have enough of this AMP, okay, this AMP will become stimulated and enable fructose 6-phosphate to get phosphorylated, and then you have fructose 1,6-biphosphate, ultimately producing pyruvate. Generally, this is occurring. Okay, pyruvate production, more pyruvate, ATP is being produced, cell respiration is occurring, ATP is being produced, and so forth and so forth. When you have enough ATP, this will come in and block phosphofructokinase. Okay, so phosphofructokinase is just an enzyme that will produce this to this, and it can be inhibited or it can be stimulated. Well, not just ATP, but also citrate can come in and inhibit this molecule. All right, so they both can be working on the regulation of this particular enzyme. Okay. Good. So the last part here that I want to mention, if the citric acid cycle intermediates are diverted to other uses, again, glycolysis will speed up. So what this means is essentially that if the intermediates, like I mentioned before, are being used to produce something else, glycolysis will speed up to replace them and to activate ATP production again. So there needs to be a balance always within the cell, and the cell does a very, very effective is very effective at doing that. Metabolic balance is also augmented by the control of other enzymes. Okay, and cells are incredibly quick at responding to changes in the environment. Incredibly quick. So cell respiration is one of the most important metabolic pathways to help, keep the cell and the organism in homeostasis. Okay, cell respiration functions in the context of energy flow and chemical cycling in ecosystems even. Just look at carbon dioxide and water, right? So the energy that keeps us alive is released, not produced, by cell respiration. And this sentence, it comes into deeper play when we talk about photosynthesis. Okay, but for now, that's all for chapter 9. We have finished the chapter. Okay, let me go into the summary pages here just for you guys to have. Okay, let me move this back here. Chapter review, please. You can pause the video so you can take a look at the summaries. Okay. Chapter review number 2. Okay, you have concepts throughout each of the sections. Let me move that there. All right. So that finishes the video, guys. Thank you for watching, and I hope this was very helpful for you. And I'll see you guys in the next one with Chapter 10 on photosynthesis. Okay, thank you very much. Take care.